Imagine flicking a molecular switch that accelerates aging in a neuron, or flips it off to reveal what drives Alzheimer's. That switch may be neddylation, a little known but powerful modifier that now sits at the center of neuronal aging research. Hello, and welcome back to Decoding Longevity, where we break down the science shaping the future of health span. I'm Raghav Sehgal, here with my co-host Macsue Jacques.
Hi everyone. In our previous episodes, we have explored how new research is tackling the rise of senescent cells as we age. And we have also discussed on new scientific models that are pushing boundaries and are transforming our ability to study age-related diseases. Today's spotlight is a comprehensive CRISPR screen that uncovers neddylation as a regulator of neuron age and Alzheimer's-like degeneration in human stem cells derived models.
To help us unpack this fascinating study, we are joined today by Nathalie Saurat, the first author of the paper and a rising star in neurodegeneration and aging research. Nathalie completed her postdoc at MSKCC in New York, working in the lab of Dr. Laurent Studer. And recently, she has started her own research group at Monash University. Her research bridges developmental neuroscience, stem cell modeling, and genome engineering to uncover mechanisms of neuronal aging and neurodegenerative diseases. Nathalie, welcome to Decoding Longevity.
Yeah, it's super excited to be here. Thanks for having me.
Thank you, Natalie. It's really, really exciting for me to have you here, especially like after having so many science discussions, informal discussions with you. But before we dig into the podcast itself and your findings in your recent paper, I just wanted to sort of like set the stage to the audience and for all of the people that are not necessarily in the science field. Could you please explain what is a CRISPR screen and how does it work and why did you choose to go down that route for your research?
Yeah, of course. So I think at this point, most people have heard of CRISPR itself, which is a super powerful technique that allows you to modulate the expression of genes. And in my case, we did this by inducing little insertions or deletions that disrupt a gene. So this allows you to kind of knock out its function. Importantly, the CRISPR screening technique was really great for neurons because it allowed me to do a whole genome CRISPR screen. So that means knocking out every single protein coding gene in the human genome, one at a time in individual neurons. And so this is super powerful. You get a huge amount of information. But as I say, to do that, you have to have a huge amount of statistical power. So you need to use a lot of neurons. And for my screen, this meant that I made 2.8 billion neurons from human stem cells, which is really a lot. And I like to give the context that, a whole mouse brain only has about 70 million neurons, as estimated recently. So that's about 40 mouse brains worth of cells. So, you have a lot of cells, but you can then, find out what is happening to the function of a lot of genes.
Yeah, that is amazing. 2.8 billion neurons. And just to think about that scale is huge in some ways. And also sort of doing CRISPR screen on all of the genes in the neurons, all protein coding genes, that's such an exciting thing to do, I would say. But I would like to go back to sort of Alzheimer's itself. So most model of Alzheimer's, as you know, sort of ignore one big factor, which is aging. Even if neurons sort of carry a mutation, they're young, they don't fully recapitulate late onset disease features. So artificially aging cells or uncovering pathways that mimic aging in some ways is kind of critical. And in that way, finding genes that regulate neuronal age in human pluripotent stem cells can reveal hidden switches, which we kind of never knew existed. Now, do you mind really expanding a bit about this, and was this what you had in mind when developing this particular project?
Yeah, so when I went into this was actually exactly the problem that was kind of eating at me. So how do we study these late onset diseases in our stem cell models? So I think when we are in the aging field, it's a little bit counterintuitive to people because most people are working with the ultimate goal of rejuvenation. But here I am trying to make my stem cell derived neurons actually older. And this is, as you touched on, when we make a cell from a pluripotent stem cell, you give them the same set of instructions that they would get during normal embryonic development. So at the end of the process, you have the right cell type, which is super important. So cortical neurons for Alzheimer's disease, dopaminergic for Parkinson's. But your cells aren't actually the right age. There's no change of time in a dish. So if you have spent a few weeks making your neurons, they're a few weeks old. And we really want to understand what happens in the brain of people with, say, Alzheimer's or Parkinson's, which takes really decades to manifest for them to see symptoms. So the idea is that if we could have cells that were the right age, we might have a lot more luck in finding new drug treatments. And even at a basic level, trying to understand why age has such a big impact on cognitive function. And of course, if you can find a way to make a cell older, there's always the hope that you can reverse this process to rejuvenate cells too.
Did you end up getting to that older cells? Did you manage to reach that point? Do you mind just
Yeah,
what
of course.
was the
So we did. In the screen, we looked at a number of different targets that came out of them. We managed to rank them according to what seemed the most interesting, and also to see what was actually happening in age in patients. And out of all of that filtering, we came up with Neddylation as this pathway that was able to drive all of these cellular aging hallmarks in our neurons, and also accelerate the formation of different Alzheimer's disease related pathologies in a dish that we couldn't actually see before without any age aspect.
Yeah. So, okay. So just following up on that question a little bit more, you just said like, okay, we screen over 2.8 billion cells and you have, all of those genes that you knocked out, knocked in. So I assume you end up with a lot of the targets, a lot of different targets. And to go through the process in selecting Neddylation itself, it would have been quite a lot of work. But I just want to sort of like go back into a little bit on the rationale that you had. Why Neddylation and why not any of the other targets that you might have come across?
The idea here is how did we kind of rank and filter our hits? And the way that we did that was actually, so the screen was done both in wild type and Alzheimer's disease neurons. So we could see which set of genes had a much bigger impact on the Alzheimer's neurons compared to the wild type. And the idea here is that if you're looking at something that's truly age-related change in an age-related disease, we would expect that the age would combine with whatever genetic vulnerability you would have, and that this would make you kind of more susceptible to having those disease phenotypes. So we were able to rank our hits based on what hits had a bigger impact on Alzheimer's disease neurons in terms of developing the Alzheimer's disease-related pathologies. And also we were able to take then this list of genes that was ranked and also filter them further based on sequencing data that we had from young and old brain, both human and mouse, and see which of those genes actually showed a decrease in expression in the old brain. So we wanted this to be as close to possible as physiological ways that the brain might be aging and that this might be affecting Alzheimer's. Does that make sense? So is it useful now to tell you a little bit about what Neddylation actually is?
Yeah, I think this is fascinating because I have done a lot of exercise studies, right? And one of the pathways that actually come up from my exercise studies is Neddylation itself. So it just sort of like showed, obviously it wasn't in the brain, it was in different tissues, but it was just showing how exercise actually also potentially have an opposite effect to aging in that specific pathway. But yeah, please tell us, what is Neddylation?
That's super interesting. We're going to need to talk a bit more about that, I think. Collaboration incoming. So one of the key pathway was Neddylation, and it's super interesting because Neddylation is actually one way in which you can tag proteins in the cell, and you tag them with this protein called Ned8. And for the scientists out there, this is a little bit analogous to ubiquitination, where you would tag your protein with ubiquitin. But in the case of Neddylation, the Ned8 changes how certain proteins behave. And in particular, Neddylation regulates this family called the Cullen ring ligases. And they actually need the Neddylation tag to be active. Through this activity, then, they are able to regulate the turnover of about 20% of the proteome. So you're actually regulating kind of proteostasis through this pathway. And this is, of course, super interesting in the context of aging, because protein turnover is impaired in aged cells. And in things like Alzheimer's disease, you get an accumulation of proteins that don't function properly. And we think that might be important to the pathology of the disease.
Yeah. And in the paper, you actually found an enzyme called UBA3, which was involved in neddylation, and also the small molecule inhibitor 4924, which led to sort of multiple senescence-like phenotypes in neurons, including enlarged nuclei, P21 accumulation, loss of LMB1, DNA damage, so on and so forth. Now, would you say that this might be a potential sort of pathway of aging Alzheimer's onset, or is there more to the story?
I don't think we know that yet. For me, I think that there's likely many ways that aging can be induced or accelerated in a cell. And I think you mentioned progerin a little bit early on, but my postdoc mentor had published a paper before I started in the lab that, for example, progerin can accelerate the onset of age-related phenotypes and Parkinson's disease phenotypes and dopaminergic neurons. So I think it's probably not a one-size-kind-of-fits-all strategy. But for me, it is a super exciting strategy nonetheless, because now that we have ways to induce aging in our neurons, we can start to study these late onset phenotypes like tau phosphorylation and aggregation. And that opens up a lot of possibilities to try and understand how Alzheimer's disease kind of progresses in ways that we couldn't before.
Yeah, that is really, really interesting. And I assume like now that you have the neurons age, not just Alzheimer's diseases, but also dementia and other age-related diseases, right? So
Yeah.
I guess this like sort of like fits into this growing movement of using stem cell models to program aging or to study aging. So let's expand a little bit more on this idea of using the stem cell for research. Where do you think this field is going? What is the potential that this emerging technique or this emerging knowledge base has to improve aging itself?
Yeah, of course. And I think this is actually a really big area of interest. I think you will have seen in the news that there's been a big push to use fewer animals in research. And I think both the EU and US funding bodies are pushing this. So this obviously fits in quite nicely with having, stem cell models that you can do more of the work in terms of studying age-related diseases in a dish. So I think that's really important practically. But also, as I said, the stem cell work's really scalable. So think back to the billions of neurons I made for the screen. Being able to work at that kind of scale when you're trying to do drug discovery for age-related disease really opens a lot of possibilities. And you can start to target possibly stages of a disease that weren't really accessible before. So as I was talking about, these kind of late stages where you're getting the tau phenotypes. In the world of Alzheimer's disease, the focus has really been on amyloid aggregation, and that's where a lot of the drug development push so far has gone. But this is the first step of the disease, and so we really want different ways with which to help patients, that are coming in when they already have symptoms. And so I think having these tools is super important because it allows you to access this phase of these late onset diseases, which are currently a bit of a black box for scientists.
Yeah, and you just mentioned about animals over there. Do you think maybe those stem cell models could be somewhat better? Because we know that one restriction that we have in research is that not always that you test something in an animal is actually translatable to humans. So do you think maybe stem cells may be a better model potentially?
I think that's been a lot of the excitement around using human stem cell models for years, is that you're really focused on the human biology and the human neuron. And animal models are amazing at a lot of things. They've really, really moved fields forward. But I think, especially with things like Alzheimer's disease, where there isn't kind of an equivalent in a normal mouse, you can give mice Alzheimer's by adding in heaps of different genes and mutations, but it's really a much more human-centered disease. And so I think it's important to work on the closest model that you have to the disease that you want to study. Now, of course, there are some things like behavioral work that are important in dementia and which are really difficult to do with stem cells. So there's always going to be a bit of a room for both, but definitely allows you to do things and access biology in a way that I think is important to these late onset neurodegenerative diseases.
So coming back to sort of neddylation, and clearly you showed the sort of the value of this as a novel axis of therapeutic intervention. And it could potentially even preserve neuronal health in aging or slow down neurodegeneration. But and the question I have is, in some ways, you have defined a really interesting sort of almost drug discovery pipeline where you can look at genetic risk, stem cell derived neurons, and aging modulation to find compounds that can potentially rescue both aging markers and disease phenotypes. Do you see both neddylation developing into a sort of new drug target of sorts, and do you see this kind of pipeline also evolving into something which might be sort of a drug discovery, target discovery pipeline for other sort of diseases? And what challenges do you see translating this from bench to bedside?
Yeah, sure. As I said, I'm really excited about this model being used as a tool for drug discovery. I think that would be super powerful, and it's some of the things that my lab is trying to do. And in addition to it being really useful in terms of looking at age-related diseases, I think you can also think about it in the context of a model to test whether strategies that target cellular rejuvenation would also enable you to kind of reverse some disease-associated phenotypes. So I think really interesting and powerful from that respect. However, what we found was that targeting neddylation itself is quite difficult because for the colon ring ligase complex that I mentioned to function correctly, it needs both cycles of neddylation and deneddylation. So it's a very dynamic process. So if you overexpress the neddylation pathway, you're going to mess up this balance. So you really want to be able to maintain it at physiological levels. However, as I mentioned, there was a reduction in this UBA3, which is the key enzyme for the neddylation pathway in old neurons. So one thing to think about might definitely be restoring it to the kind of level that it's normally at in young. So this is definitely something that we've thought about and could be interesting going forward.
I guess you can say threading the needle with the neddylation pathway is difficult. Sorry, that was a bad joke. But thank you, Natalie, for this incredible work and sort of sharing your insights about this really interesting paper. And again, congratulations to you and all the other authors for this landmark study. And thank you for being here.
My pleasure. And it's been super exciting to try and find ways to program age into neurons, and I'm really excited by seeing how these models get used going forward. Thanks for discussing this with me today.
Thank you so much, Natalie. And if you enjoyed today's episode, check out the paper. We will put the link again in the show notes. And don't forget to subscribe, rate, share with whoever you think may be interested in aging, biology, and research. We will keep bringing up new episodes and guest speakers. So make sure you tune in for the next time.
See you all next time. Thank you.